Now we will discuss about RNA interference. This is a very important topic of RNA biology, and this procedure will also help us a lot of aspects relating to biotechnology, and this process is also very much important for plant also, for plant biology also. Let us discuss this. What this RNA interference means actually. Let us first simply understand, then we will go to detailed procedure. Okay? Interference, what interference means? RNA interference is also written as RNAi. What interference means? Interference means दूसरों का काम पे दखल देना, दूसरों का काम पे दखल देना, किसी को उंगली करना, किसी दूसरे का काम को बिगड़ना. These are called interference. Okay? So RNA will interfere, one question, or RNA will be interfere. Which is correct here? Let us see it. Both option is correct. In this procedure, RNA will be interfered and RNA also interfere. How? Let us see. There is a lot of type of RNA, you know. How one gene expressed, you also know. Suppose this is one piece of DNA. We are taking that this is one gene. This gene is expressed as mRNA. This is called transcription, you all know. Then this mRNA will then translate. It will take part in translation and it will produce polypeptide chain. This is actually the central dogma, you all know, and in this way gene is expressed. So now let us get into the topic RNA interference. Here, which type of RNA will be interfered? Which type of RNA will be victim? A lot of RNA types is there: mRNA, tRNA, rRNA. RNA interference don't deal with those all. It deals only with the most important RNA, mRNA, means messenger RNA. This RNA will be interfered, means this RNA will be the victim. So let us write down here, this is the victim. Who will interfere? Interestingly, that is also one RNA. Who will interfere? RNA. And that is not one normal RNA, you know. RNA will not interfere normal RNA because they all are RNA, they are friends. But one absurd type of RNA. RNA is generally single-stranded in our body. But suppose in eukaryotic organisms there is any double-stranded RNA somehow has produced. We will discuss how that will be produced. That double-stranded RNA will interfere mRNA. So who will interfere? Double-stranded RNA, dsRNA. So dsRNA will interfere mRNA. How mRNA will be interfered? Let us see it. If suppose this is the mRNA, then what is basically in the mRNA? mRNA is a polynucleotide chain. It has one structure, and another one important thing is that mRNA does translation. mRNA expresses itself through translation and it forms polypeptide. Means it has one structure and function. It has structure, any molecule has structure as well as function. This double-stranded RNA, which may be produced inside the cell or which may be alien one, which may come into a cell due to many other exogenous reasons. Its production may be endogenous, may be exogenous. We will discuss it. But here, look at, in this RNA interference, this double-stranded RNA will interfere both in the structure and function of the normal mRNA. So if the normal mRNA's function is disturbed, means translation will not occur. If translation will not occur, there will be no polypeptide. If there is no polypeptide, then we can say that there is gene, there is mRNA, but there is no polypeptide. But our ultimate aim from or ultimate expectation from a gene is what? To get one polypeptide product. But if there is no polypeptide, then we can say that the gene is silenced. So RNA interference is actually one gene silencing procedure. What it is? It is actually one type of gene silencing procedure, silencing method, you can say, by which? Through RNA. So it is in which level? It will be in the translation level, means it will act on mRNA. So it is RNA mediated gene silencing method. And this method is a very common method. Where it occurs? It is a very common method. It occurs in all eukaryotic cells. It occurs in all eukaryotic cells. So where it occurs? First of all, let us write down. Occurs in all eukaryotic cell. It occurs in all eukaryotic cell. But let us see. This is actually silencing one gene. So why this will go to happen? This will go to occur inside one normal cell. What is function? It is not a normal procedure. Actually, it occurs in all eukaryotic cell, but it occurs as a method of, as a process of cellular defense. Okay, it is not a normal procedure, a normal metabolism. It is not a part of normal metabolism of one eukaryotic cell, but it happens as a cellular defense. Cellular defense. So what is RNA interference is? RNA interference is one type of procedure where mRNA will be affected, will be silenced, and this occurs in all eukaryotic cell as a method of cellular defense. Let us get into the inside of this topic. Let us discuss about the procedure of RNA interference. What we have told already? We have told that RNA interference is mainly caused by one double-stranded RNA. One double-stranded RNA. Suppose this is the double-stranded RNA. This is double-stranded RNA, dsRNA. So to initiate RNA interference, we need one double-stranded RNA inside a cell. Suppose this is a cell. There we need double-stranded RNA. But normally this double-stranded RNA is not very common inside the cell. So what would be its source? How this double-stranded will be produced or generated inside a cell? It may be endogenously produced or it may be exogenously produced. So how will it come here? It can come endogenously or it can be formed outside as exogenous, and it may be also produced inside the cell as endogenous. So it can be exogenous, or it can be endogenous. Okay. So what are the exogenous methods or procedures by which double-stranded RNA is formed inside one cell? Let us discuss this. Exogenous. Number one. So we are discussing about exogenous origin. Number one is viral infection. Viral infection having RNA genome. Suppose one virus has attacked this, and that virus is RNA virus. In this way, double-stranded RNA may be introduced inside one cell. Number two is laboratory manipulation. You can artificially synthesize one double-stranded RNA in the laboratory, and you can introduce that into that cell using some vectors. You know, if it is plant and it is generally used for plant, then agrobacterium vector may be used. So number two is laboratory manipulation In laboratory manipulation, what you will do? You will create one double-stranded RNA artificially, one dsRNA artificially. You will then introduce it using some vector. Okay. Number 3, you will do the same thing, laboratory manipulation, but you will not introduce one double-stranded RNA there. Instead of that, you will introduce there one double-stranded DNA, one dsDNA. This dsDNA will be introduced by using vector. But this DNA will be special type of DNA. After going inside the cell, you know, there is 2 strands of DNA, 5 prime to 3 prime and 3 prime to 5 prime. And during transcription, only one strand is transcripted. Both the strand is not transcripted. But this DNA will be specially designed so that it will transcribe both its sense and antisense strand. So if this strand 3 prime to 5 prime, these will form these RNA, and suppose this is one from here, this is the normal sense RNA, okay, and this will form antisense RNA. So this antisense RNA will be complementary to the sense RNA, and obviously they will just simply add and will form one dsRNA. So these are the exogenous sources by which one double-stranded RNA may be introduced, may be produced, may be generated inside one cell. One is viral infection, number 2 laboratory manipulation of double-stranded RNA or laboratory manipulation of double-stranded DNA and inserting them inside the cell using one vector. Okay, let us now see what are the endogenous, how endogenously double-stranded RNA may be produced inside one cell. This was our cell. Suppose this is the nucleus. In some cells, there are generally some double-stranded RNA is present inside the nucleus. Generally, some are present. Some generally double-stranded DNA may be present inside that cell. Number 1, and number 2. So number 1 may be some dsRNA may be naturally present. May be naturally present. So if it is naturally present, then one question will arise in our mind, how it has been formed. The answer is very simple. Somehow, some part of the original genome, some part of the DNA has bidirectionally transcripted. Both the strand has been transcripted, so that sense and antisense both RNA has been introduced and they has attached to each other as they are complementary to each other. In this way, some dsRNA may be naturally present inside the cell. Number 2, some double-stranded RNA may be produced by some mobile genetic elements. Mobile genetic element means transposons. Okay, it may be produced by mobile genetic element or elements. Mobile genetic elements is normally transposons. But these transposons are not normal. These mobile genetic elements are not normal. Okay, these are such type of mobile genetic element which replicate via an RNA intermediate. And this type of transposon will be called as retrotransposon. What it will be called? Retrotransposons. Retrotransposons. So some double-stranded DNA may be formed, or dsDNA may be formed inside the cell by retrotransposons. Let us see what is the mechanism actually. Let us see the mechanism. Suppose this is the DNA, okay, this is DNA, and this part is transposon. Transposon means this is jumping gene. This can copy itself, this can copy and paste itself at anywhere in the genome, and it causes mutation also. Suppose this is the transposon, and this is obviously DNA, part of DNA, part of the genetic material. This will transcript itself into RNA. Then you hear the word retro. You have already heard the word retro. So what retroviruses do? Retroviruses do reverse transcription. They have the capability to form DNA from RNA, and that same capability, the same procedure will be followed by retrotransposons. Here retrotransposon will form this RNA to DNA, this RNA to DNA, and this DNA will be pasted here. So remember, this is retrotransposon present in eukaryotic cell, but it has the property of one retrovirus, that is HIV actually. So these are the internal sources or endogenous sources by which double-stranded RNA may be produced inside one cell. So now let us discuss more detail the procedure of RNA interference. So double-stranded RNA is ready right now. It is double-stranded RNA. Where it has been produced? Suppose this is the cell. This can be produced both inside the nucleus, or it can be produced in the cytoplasm. If it is cytoplasm, then there is no problem. But if it is in the nucleus, then this is to be immediately transported, and it is transported through the nuclear pore of the nuclear membrane, and it will come into cytoplasm, and the actual procedure will occur in the cytoplasm. So what will happen? There is one type of special ribonuclease protein. In each and every eukaryotic cell, there is one type of special ribonucleoprotein. This protein is called dsR. What it is called? dsR. So now this dsR protein will act on this. This is dsR. This is what? This is dsR. This dsR is actually RNase 3 type of enzyme. This is one RNase 3 type of enzyme. This is RNase 3 type of enzyme. That means it will cleave double-stranded RNA into small fragments. And after the cleaving activity of dsR, this double-stranded RNA will be fragmented into small pieces, and different type of fragments will be produced. Basically 3 type of fragments are produced. One is called miRNA, another one is called shRNA, another one is called siRNA. Here mi means microRNA, here sh means short hairpin RNA, si means small interfering RNA, So what has happened? Double-stranded RNA, dsR acts on it, and dsR chop up it into small fragments. These fragments may be 20 to 25 base pair long, 20 to 25 base pair long. Among these fragments, we will take this siRNA. As we have. miRNA also does it, shRNA also does it, but the procedure is different. We will discuss only the method which is used by siRNA. Okay, so this siRNA is small fragment 20 to 25 base pair long. This is the siRNA, and suppose this is 3 prime. Suppose this is 5 prime to 3 prime, and this is 3 prime to 5 prime. Okay, and in each and every wire 2 nucleotide will be hanging. 2 nucleotide stretch will be empty each and everywhere at the 3 prime end. 1, 2. So this is small siRNA. Now this siRNA, what it will do, this siRNA will now add with some protein complex. It will add with some protein complex. It will add with some protein complex, with some proteins. Some proteins like argonaute, slicer. These are some proteins which will add with siRNA and they will form one complex. Okay, they will form one complex. So right now this siRNA is not alone. It is with some proteins. It is with some proteins. With disher protein. Sorry, with slicer and argonaute protein. Proteins. So this complex, where there is one siRNA and some proteins, argonaute, slicer, etc., this complex is called RISC. RISC. RNA induced silencing complex. What it is? RNA induced silencing complex, RISC. So in this RISC, means RNA induced silencing complex, what will happen? That is very interesting. You know, there is actually two strands. These two strands will not remain after the activity of these enzymes argonaute. What will happen in the next stage? The argonaute will cleave and will chop off one among the two strands. One strands will be called as a passenger strand, and another one will be called as guide strand. This passenger strand will be completely destroyed, will be completely chopped off and completely cleaved by the argonaute. So right now what we have one single stranded guide RNA, okay, and this complex we have right now this. Now what will happen? This is guide RNA, okay. This guide RNA will help this complex RISC to find the target mRNA. Suppose this is the target mRNA, okay. This is the target mRNA. Now this guide RNA will go to this target RNA and it will join here. How it will join? Based on complementarity. Actually this guide RNA is complementary to the target RNA. Which RNA is to be destroyed or which RNA is to be interfered, that RNA's nucleotide sequence is just complementary to the guide RNA. So now this guide RNA will add with this. Suppose this is the full RNA. The total RNA will not be added, only the small fragments equal to the guide RNA will be attached, as these are complementary to each other. Now this argonaute protein is present there. This argonaute protein, what will it do? It will simply make the mRNA into small fragments. So now this mRNA will be some small fragments, okay. So what will be the fate of these small fragments of mRNA? So obviously mRNA is destroyed, then there will be no question of translation, no chance of translation, as the mRNA has been fully destroyed and it has been chopped up and cleaved by the argonaute. Now this mRNA will soon be detected by the cell's defense as non-essential substances, and these will be destroyed very soon by the cellular defense. So in this way RNA interference totally silence one mRNA or one gene. This is RNA interference.